Hey chams of Examsnet, welcome to the question series of Examsnet. So I hope this series of detailed explanations of the previous year's questions of NEET is helping you a lot. So we are again, going to deal with a total of 100 questions, which is of NEET 2022 examination that has been asked. So let's start with this session now. So the very first question is from the botany sections that we're going to deal with. And the first question says that given below are the two statements that has been given. All right. And out of the two statements, you have to choose the correct statement from the options given below. Now, let me tell you that the statement one says that sickle cell anemia and hemophilia, they are autosomal dominant traits, while the statement two says that sickle cell anemia and hemophilia are disorders of the blood. All right, so this question has been asked from the chapter of your principle of inheritance and variation of standard 12. And let me tell you that this statement, the first statement is considered to be as the incorrect. So the statement first is actually incorrect because the reason is that sickle cell anemia and hemophilia, these are both are considered to be as the recessive trait. The only difference is that the sickle cell anemia is an autosomal recessive trait or a disorder, while the hemophilia is an X-linked recessive trait or disorder. All right. So we can say that sickle cell anemia is an autosomal recessive disorder. All right. While hemophilia, you can say, and hemophilia is X-linked recessive disorder. X-linked recessive disorder, which is more common in men as compared to women. All right. So this is the correct statement. All right. That means this is an X-linked recessive disorder and sickle cell anemia is an autosomal recessive disorder. And yes, the statement second, which says that sickle cell anemia and hemophilia, they are the disorders that are related to blood. So this is again a true statement. This is a correct statement because both of the disorders are related to blood. So this is basically the statement two is correct because sickle cell anemia is one such disorder in which the shape of the RBCs changes from biconcave shape to sickle shape. That's why. So statement two is correct. Statement two is considered to be correct because we can say both are blood related disorders. All right. Now the reason is that in sickle cell anemia, the shape of the RBCs, it changes. The shape of RBCs changes from biconcave to sickle shaped. That means it's a kind of a boat shaped structure. So the biconcave structure is going to change into sickle shape. And because of that, the retention of the blood in the RBCs is very less. So it changes from biconcave to sickle shape, and it is because of the substitution of a very important amino acid, which is glutamic acid by valine. So you can say due to substitution of due to substitution of glutamic acid. Glutamic acid is an amino acid by valine at sixth position of beta globin chain. Sixth position of beta globin chain of hemoglobin. So because of this reason, the shape changes, all right. And in case of hemophilia, hemophilia is basically considered to be as a, this is a blood clotting disorder, basically. All right, so hemophilia is a blood clotting disorder. And here basically all the cascade of proteins which are involved in the clotting process of blood, that gets affected, and because of that there is excessive bleeding. So where cascade of proteins involved in blood clotting gets affected, and so there is excessive bleeding. So this ultimately results into excessive bleeding, even if there is a small cut in the body, in any part of the body. All right, so this is the reason. So that's why we can say that the statement 1 is incorrect and the statement 2 is correct. So out of all the options that you can see, the first option which says that statement 1 is incorrect but statement 2 is correct, this is the correct answer. That means option A is the right answer. All right. Now question number 2. Question number 2 says that which stage of meiosis can last for months or years in the oocytes of some vertebrates? Dikinesis, leptotene, pectine, and diplotin. Now, this question has been asked from the chapter of your. This is a very generalized question that has been including the knowledge of that includes the knowledge of both the chapter from the cell cycle and cell division, and also from your reproduction part as well. So let me tell you that out of all the stages, it is basically the diplotin stage that can survive for months or years. It is this stage. Basically, there are 5 stages of meiosis: one, leptotene, zygotene, pectine, diplotin, and dikinesis. So diplotin is the stage that can remain suspended for months or years, you can say. So we can say that in oocytes of some vertebrates, oocytes of some vertebrates, diplotin lasts for months or years. This basically lasts for months or years, and that's why this is considered to be as suspended diplotin stage. So that is suspended diplotin stage. All right. So diplotin is one such stage where the recombination has taken place, and this recombination, the result of the crossing over or recombination, which is basically the products of the recombination, they are seen. So once the products are formed, they are stabilized. All right. So that's why it can remain for months and years, and it can last up to months and years in the oocytes of some vertebrates. So that's why option D is the correct answer. All right. While in the case of this dikinesis, leptotene and pecitine, these are all the processes which is going on. These are the stages in which the process is still going on. But diplotene is one such stage where the process has ultimately resulted into one product has been formed, which is the crossover product. All right, and that is called chiasma. So that is the case. So that's why option D is the correct answer. Now, question number 3. Now, question number 3, which says that given below are the two statements. One is labeled as assertion and the other is labeled as reason. So, assertion says that when a particular restriction enzyme cuts the strand of DNA, the overhanging stretches or sticky ends are formed. All right. So, some restriction enzymes cut the reason for this assertion is that some restriction enzymes cuts the strand of DNA a little away from the center of the palindromic site. So a little away from the center of the palindromic site. So in the light of the above statements, you have to choose the correct answer from the given options that is below. All right. Now, this question has been asked from the chapter of your biotechnology principles and processes of your standard 12. All right. So if you just look into the assertion and reason statement, so both of them, that means the statements, the assertion also and the reason also, that is going to be the correct one. And yes, reason is actually the R is actually the correct explanation of A. So basically, option B is the correct answer, and the reason for this is because when a restriction enzyme is going to cut a DNA strand, the overhanging stretches or the sticky ends are formed. Because until and unless the sticky ends are formed, the two DNA cannot be combined with one another. So we can say that both A and R are correct, and R is the correct explanation of A. It's the correct explanation of A. All right. So how we are going to justify this? Because whenever a particular restriction enzyme, so we can say that whenever a restriction enzyme let us consider with an example like ECO R1. All right, so this is this one. So whenever a restriction enzyme like this one, cuts the DNA strands, and that to both strands. That's why I have written strands of DNA a little away from the center of palindromic sites, the sticky ends are produced. Because these sticky ends will be ultimately joined by the DNA ligase. All right, but it should be between the same two bases of the opposite strand. But this excision I'm talking about actually, say the sticky ends are produced. Now let us consider, for example, we can take an example of a palindromic sequence which is being cut by EcoRI. So this is just an example that I'm drawing, writing it up. So this is the case. All right. So if you will see the action of EcoRI, so it is going to cut over here. So EcoRI is going to cut over both the strands over here first. All right. So that is the case. So that's how it is going to create the sticky ends. And the next statement, which is saying that some restriction enzymes cuts the DNA strand a little away from the center of the palindromic sites. Yes, when the cutting is done from the center of the palindromic, because this is a palindromic sequence. This is the palindromic sequence that I have palindrome sequence. You can say that I have written. And this is a very specific sequence which is being cut only and only by this enzyme, which is produced by E. coli and not by any other restriction enzyme. So that's why both of them are considered to be as correct. All right, so option B is the correct answer. Now, question number 4. Question number 4 says that give the correct descending order of the organism with reference to their estimated number found in the Amazon forest. Plants, invertebrate fishes, birds, mammals. Choose the correct answer from the options given below. Now, this question has been asked from the chapter of your biodiversity and its conservation from your standard 12. And let me tell you that with the estimation that has been found in the Amazon forest, out of all the options that has been given, insects in the vertebrates, the insects occupy the maximum population, which is approximately around 70%. All right, so we can say the B option will be the very first option, which includes insects that comes under invertebrates. And their total estimates that are found in the Amazon forest is approximately 2.5 million species. 2.5 million species. Now, if you just after this insects, the greatest number is that of the, in case of vertebrates, the greatest number is that of the plant category. All right. So the next is definitely your plants. That means which counts for approximately 50,000 species. Then after plants, the next category in the animals to be in the vertebrate are the fishes. So then comes the fishes category. So this will count for 3,000 species. Then comes the bird, I guess. D. Sorry, it's E. E. So then comes the birds. And the last will come the mammals. That means D. So birds have a count for approximately 1,300 species. That means 1,300 species. And in mammals are approximately found 430 different kinds of species are found in case of the Amazon forest. So if you look into the sequence, so the descending sequence will be, or descending order will be basically descending order. That is going to be B, then A, then C, then E, and then D. This is the one. All right. So let us take a look into the options that has been given. So the first option is B, A, C, E, D. B, A, C, E, D. Okay. So the first option only has turned out to be the correct one. So option A is your correct answer. All right. Now, question number 5. Question number 5 says that in lacoperon, the Z gene codes for transacetylase, beta-galactosidase, permease, repressor. All right. Now, let me tell you that this question has been asked from the chapter of your molecular basis of inheritance of your standard 12. And lacoperon, in case of the lacoperon, there are 3 structural genes which are prominently known. All right. So in lacoperon, There are 3 structural genes that are known. So 3 structural genes known. And those 3 structural genes are Z, Y, and A. All right. So Z gene, it is the Z gene that codes for or encodes for beta galactosidase. It's beta galactosidase, which is going to act on the lactose, thereby converting it into glucose and galactose. So this encodes for beta galactosidase. Y basically encodes for enzyme permease. So this codes for permease. So permease is basically responsible for increasing the permeability of cell for lactose. All right. It increases the cell wall permeability for lactose, so that lactose can easily enter inside the bacterial cells. While A codes for transacetylase. So this codes for transacetylase. All right. So it has been here asked about the Z gene. So Z gene is basically encoding for option B, which is beta galactosidase. All right. So this beta galactosidase, as I told you, that this is basically responsible for converting lactose. Basically, beta galactosidase acts over here to convert lactose into glucose and galactose. All right. So this is how it is responsible. While repressor is a protein which acts on the operator region of the repressor of the lac operon, and thereby it inhibits the functionality of the structural gene. So this has nothing, no relation to do with that of the encoding part, that means the structural gene parts. Now, question number 6. Question number 6 says that the 5-carbon compound formed during the TCA cycle, 5-carbon compound it is asking about, formed during the tricarboxylic acid cycle is fumaric acid, alpha ketoglutric acid, oxalosuccinic acid, and succinic acid. All right. So let me tell you that this question has been asked from the chapter of your respiration in plants of your standard 11. And TCA cycle, tricarboxylic acid cycle, is basically the citric acid cycle. All right. So TCA cycle is citric acid cycle. It is citric acid cycle or tricarboxylic acid cycle, also known as tricarboxylic acid cycle, basically. All right. Now in this cycle, the only 5-carbon compound which is formed is the alpha ketoglutarate. It is a very direct question that has been asked because every compound has its own carbon number. So it is the alpha ketoglutarate. Alpha ketoglutric acid, you can say. Is a 5-carbon compound. All right. In TCA cycle. And regarding the other components that has been given, like fumaric acid is a 4-carbon compound. Fumaric acid and succinic acid, both these are 4-carbon compounds. These are 4-carbon compounds. This is also 4-carbon. This is also 4-carbon. All right, which is formed as a result of oxidative decarboxylation of alpha ketoglutric acid. While oxalosuccinic acid is an example of 6-carbon compound. So this is a 6-carbon molecule. All right. So definitely the answer is option B over here. Now, question number 7. Question number 7 says that in meiosis, the crossing over and exchange of material between the homologous chromosome catalyzed by the enzyme polymerase, phosphorylase, recombinase, and transferase. Again, this question has been asked from the chapter of your cell cycle and cell division of your standard 11th. And this is a very direct question that has been asked just regarding the name of the enzyme. All right. And let me tell you that crossing over, the process of crossing over and the exchange of genetic material that takes place between the two homologous chromosomes. Like, for example, these are the two chromosomes over here, and the crossing over is taking place over like this. All right, so this is the exchange of the genetic material which is taking place over here. All right, so this process of crossing over between two homologous chromosomes is catalyzed by recombinase enzyme. Because since the process of recombination is taking place, so definitely the enzyme involved will be recombinase. All right, so here the crossing over is catalyzed by the recombinase enzyme. That means option C is the correct answer over here. While polymerase is an enzyme which is involved in DNA replication. So this is involved in DNA replication. All right. DNA polymerase or RNA polymerase, whatever that will be the polymerase. Phosphorylase is an enzyme that is basically involved in ATP synthesis. So this deals with ATP synthesis. And transferase is an enzyme that catalyzes transferring of a group from one to another. So this catalyzes a group transfer from one to another. One to another molecule. All right, so that's why the correct answer is option C. Now, question number 8. Question number 8 says that all successions, irrespective of the habitat, proceed to which type of climax community? Edaphic, xeric, mesic, hydrophytic. Now, this question has been asked from the chapter of your ecosystem of your standard 12. And let me tell you that any type of community or any type of succession, whether it's a, in your course, there are two types of successions that you people might have studied as well. One is called as the hydrophytic succession and the other is called as the Xerarch succession. So any succession, like for example, if there is a Hydrarch succession, all right, or if there is a Xerarch succession. There are two types of succession that you might have studied in your course. So Hydrarch succession is the succession that starts from wet areas. All right, so that includes ponds, lakes, or anything. While this is the succession which is going to start from dry areas. So dry areas basically includes rocks or something, rocks or sand or something. That is the case. So any kind of succession will lead into a a kind of a climax community, and that climax community usually has a mesic kind of a condition. So this leads to climax community, which is in an equilibrium condition, and this climax community has mesic condition. So this basically has mesic condition, and mesic condition means medium water condition. So this implies medium water conditions. Medium water conditions, in other words, we can say as neither too dry nor too wet. So that is neither too dry nor too wet. So this is known as medium water. Any of the succession, if it will be starting. of wrong. All right. So that's why it is mesic. Option C is the correct answer. Xeric cannot be the case. Hydrophytic cannot be the case because they are the origin points of succession. And edaphic, it is related with soil factors. So again, this is also not the correct answer. So that's why option C is the correct answer. Now, question number 9 over here. The question number 9 says when a carrier protein facilitates the movement of two molecules across the membrane in the same direction, so that is called as. All right. So symport, uniport, transport, and antiport. Now, this question has been asked from the chapter of your transport in plants of your standard 11. Now, let me draw a simple figure to make you people understand that what it exactly means. We can like draw a simple figure to explain this part. Right. So basically, there are three types of transportation which is being involved. All right. Three types of transportations are there. So the one is called as uniport. The one is called as antiport. And the other is called as symport. All right. So this is basically the membrane which you can see. And this is basically the membrane protein which you can see over here. Protein. All right. So whenever there are two molecules involved. Whenever there will be two molecules that will be moving in the same direction, as you can see over here, in the first figure, this is the first figure, this is the first category, this is the second category, and this is the third category. All right. So this is A, this is B over here, and this is A. All right. So whenever two molecules will be moving in the same direction, all right, so that is considered to be as symport. This is considered to be as symport. And whenever the two molecules, they will be passing the membrane protein, but in an opposite direction, that will be called as antiport. And when a single molecule will be passing the membrane protein or across the membrane with the help of a membrane protein, irrespective of the presence of the other member. So that is called as uniport. So this is called uniport. All right. So let us consider, let us consider what it has been written. When a carrier protein facilitates the movement of two molecules across the same direction. So the two molecules are moving that in the same direction. So that's why symport. So that means this one. So two molecules moving in same direction. So this is considered to be as the correct answer. So that means option A is the correct answer. Now, question number 10. Question number 10 says that when one CO2 molecule is fixed as one molecule of triose phosphate, which of the following photochemically made high energy chemical intermediates are used in the reduction phase? All right. See, this question has been asked from the chapter of your photosynthesis in higher plants, and that too from your 11th standard. All right. So, and this question is particularly from the C3 cycle, basically. All right. And whenever one CO2 molecule is being used for fixing one molecule of triose phosphate, so it is asking here about the high energy chemical intermediates that are used in the reduction phase. So for reducing, the two things are required. One is the ATP and the other is the NADPH. So if there is one molecule of CO2 which is getting reduced to one, definitely one triose phosphate, one molecule of triose phosphate. So definitely two ATPs and two NADPH are required. All right. So we can say that one, or rather we can say for fixing one CO2 molecule into one molecule of triose phosphate. So triose phosphate here basically means glyceryl dihydrophosphate. Triose phosphate, which is glyceryl dihydrophosphate, shortly termed as G3P. So a total of 2 ATP and 2 NADPH is required. All right. So this is the case. So again, this is a very direct question that has been asked, because in the reduction phase, the molecules that are required are 2 ATP and 2 NADP, and that is, it is very in a successive manner. All right, because this CO2 will be ultimately accepted by the RUBP in the presence of ribisco enzyme, and then it will be further cleaved to give rise to bisphosphoglycerate, then 3-phosphoglycerate, and that's how the process goes. So option A, if you look into the options, option A is the correct answer. The next question is question number 11. So this question states that the ability of plants to follow different pathways in response to environment leading to formation of different kinds of structures is called. All right. So is it differentiation, redifferentiation, development, and plasticity? Now, this question has been asked from the chapter of your Plan, Growth, and Development of Standard 11th. And let me tell you, this is a very direct question that has been given. So the phenomenon by which a plant will be showing different kinds of structures, so this phenomenon is considered to be as plasticity. All right. So plasticity is a process by which a plant adapts different kinds of structures. It's a process by which plants shows different kinds of structures. Now, different kind of structures means what? Different kind of structures means like changes in their morphology of the plant body, changes in the external structure of the plant body. So for example, we can say like heterophily. Heterophily is one of the example of plasticity, where plants, they usually show different types of leaf morphology, like heterophily, a phenomenon in which plant forms different leaf structures. Leaf structures, right? So they are going to show different kinds of structures like heterophily in response to the different environments, or in response to environmental adaptation. In response to environmental adaptation. So that's why the correct answer is plasticity. Now, regarding the other three definitions which have been given, so if you talk about differentiation, so differentiation is basically a process in which a particular cell or a tissue which is being involved, that loses the process of cell division. All right. So this is basically a mature cell. This is differentiation, basically indicates the maturity of a cell. So differentiation is a process in which a cell loses the capacity of cell division. So cell is going to lose the capacity of cell division. Cell division. And acquires a specific function and shape. And acquires specific function and shape. All right, so this is about the differentiation that we have seen. Now regarding redifferentiation, if I tell you, then redifferentiation is a process in which the cell, it's a process in which that comes after dedifferentiation. So it's a process coming after dedifferentiation. Process that comes after dedifferentiation, where a cell again regains the ability of division, will now lose the ability of division. All right, so that is the case. And the last is development. So development is a process, or it's a combination of growth along with differentiation. So we can say this is a combination of growth along with differentiation. So this is considered to be as a development. All right, so that's why the correct answer is option D over here. Next is question number 12. So question number 12 is a match the column kind of a question, and you have to choose the correct answer from the options given below. All right, now let us put the options. So Chlamydomonas. So Chlamydomonas is basically a green alga. So Chlamydomonas is an alga, and that too it's a green alga actually. All right. Then option B, that means this is going to match up with A. Then Cycas is a gymnospermic plant species, so this is going to match up with B. So Cycas is gymnosperm. Then option C is Selaginella, which is a pteridophyte. So this is C. Selaginella is a pteridophyte. Heterosporous pteridophyte. And option D is the sphagnum, which is a moss category. So sphagnum, which is a moss category. All right, so let us take a look into the options that has been given. So A is going to match up with third option. Okay. Then B is going to match up with fourth option. That means this one. C is going to match up with second. And then D is going to match up with first. So that means option C is the correct answer. All right. So that is the case. Now, question number 13. Question number 13 says that interfascicular cambium is present between. All right. So basically, there are two types of cambium. One is called as the interfascicular cambium, and the other is called as the intrafascicular cambium. All right. So let me tell you about both the cases. So if, for example, we can just draw a figure to explain this. So suppose these are the vascular bundles which are arranged in the form of ring like this. These are arranged like this. And the vascular bundles arranged in the circle. All right. So the lower part is the xylem part. This one belongs to xylem. While this one is the phloem part. So this is basically the vascular bundle that I have drawn. This one is the pith part. All right. Now, two types of cambium are there. One is considered to be as I told you, intravesicular cambium and intervesicular cambium. So, intravesicular cambium is the cambium which is present within the vascular bundle. So, cambium present within vascular bundle. All right. So if I just draw like this, for example, this is a vascular bundle and this is the cambium. All right. So here lower part is the xylem part and this one is the phloem. So this is the phloem. This is the cambium. That means intravesicular cambium. And this is the xylem. All right. So this is basically the intravesicular cambium that we have drawn. While intervesicular cambium, as you can see over here, this vesicular cambium is present within the vascular bundle, between two vascular bundles actually. So this ring that you can see, which is formed, this is the intervesicular cambium. This is intervesicular cambium. All right. So that means the ring which is formed over here. So intervesicular cambium is the cambium present between two vascular bundles. So that is known as intervesicular cambium. All right. So you can see over here, it is present between two vascular. This is the vascular bundle and this is the vascular bundle. So it is present within the two vascular. All right. So let us take a look. Secondary xylem and secondary phloem, no. Primary xylem and primary phloem, no. Pericycle and endodermis, two vascular bundles, no. Then between two vascular bundles, this is true. So that means option D is the correct answer. Now, question number 14. Question number 14 says that which of the following growth regulator is an adenine derivative? All right. Now, let us just talk about this one. That means abscisic acid, auxin, cytokinin, and ethylene. So this question is asked from the chapter of your planned growth and development of standard 11. So the sources, if you recall the sources where these growth regulators are have their origin. So ABA is one such hormone. That means abscisic acid is one such hormone which is having a carotenoid derivative. So this has carotenoid derivative. Carotenoid derivative. All right. Then auxin is having the derivative of auxin is basically considered to be as indole compounds. All right. So auxin have indole derivative compounds. Indole derivative. All right. Then cytokinin. In case of cytokinin, specifically the kinetin, if you take a look. So in case of cytokinin, the kinetin, which is a synthetic cytokinin, you can say. Synthetic cytokinin, you can say. So this kinetin is basically having the adenine derivative. This is having adenine derivative, and this adenine derivative is called as N6 perpuryl aminopurine derivative. Right. So we have found out our answer. While the D1, which is the ethylene, ethylene is a gaseous form. So this is a gaseous form. So that means option C is considered to be as the correct answer. All right. Now, question number 15. Question number 15 says that the chromosomal theory of inheritance was proposed by. All right. So the chromosomal theory of inheritance was proposed by Robert Brown, Thomas Morgan, Sutton and Bowry, and Gregor Mendel. All right. Now this question has been asked from the chapter of your principle of inheritance and variation of standard 12. And it's a very, very direct question that has been asked. So let me tell you that this theory of chromosomal theory of inheritance, the chromosomal theory of inheritance, this was proposed by Sutton and Bowell. Or rather, we can write the full name. It was just proposed by Walter Sutton and Theodore Bowley. Walter Sutton and Theodore Bowley. And that too, the year something like this, 922, 1903. All right. Regarding the other discoverers' names that has been given over here. So Robert Brown, he basically discovered nucleus. So he's the discoverer of nucleus. So he discovered nucleus. Then Thomas Hunt Morgan, he gave the concept of linkage inheritance. So gave concept of linkage and inheritance. All right. Then Sutton and Bowry, as it has been already been clear. And Gregor Mendel, he gave the principle of inheritance. He gave the concept of principles of inheritance. So gave principles of inheritance. All right. So that's why the correct answer is option C over here. Now, question number 16. Question number 16 says that which of the following statement is not correct? All right. So which of the following statement is not correct? The rhizome is thick, prostrate, and branched. All right. Rhizome is a condensed form of stem. The apical bud in rhizome always remains above the ground. The rhizome is aerial with no distinct nodes and internodes. So this is a very generalized question that has been asked regarding the concept of ribosome only. All right, and let me tell you that not correct it is asking about, that means that which is a false statement, we have to go for it. All right, so if you talk about rhizomes, so rhizomes are those structures which are considered to be as an underground stem modification. So rhizomes are considered, this is a question that has been asked from the chapter of morphology of flowering plants, because the modification of their modifications has been given. So from that section, so rhizome is basically considered to be as an underground stem with having, having the presence of distinct nodes and internodes. All right, so definitely we can say that the incorrect statement is option D over here, because the correct one is that the rhizome is an underground stem modification. So this is basically an underground stem modification having distinct nodes and internodes. And like one of the best example, if you say, then there is ginger, then colocasia. These are the examples of rhizome. So definitely the rhizome is thick, prostrate, and branched. This is true. Rhizome is a condensed form of stem. True. This is also true. The apical bud in rhizome always remains above the ground. All right. Yes, because that apical bud has leafy shoots. That's why. So this is also true, and this is the wrong option. So according to the question that has been given, so the incorrect statement, not correct statement, it is asking about. The incorrect statement is option D over here. All right. Now, question number 17. Question number 17 says that the phenomenon by which the undividing parenchyma cell starts to divide mitotically during the plant tissue culture is known as. All right. So this question has been asked from the chapter of your plant growth and development. And again, this is a very direct kind of a question that has been asked. So see, there are basically three processes. All right. So differentiation, dedifferentiation, and redifferentiation. So differentiation is one such process in which the cells loses the ability of division. So during differentiation, cell will lose the ability of division. Cells loses ability of cell division. So cell will lose the ability of cell division and matures. And that cell is going to ultimately mature. All right. Now, after the differentiation, after the differentiation process, then comes the dedifferentiation. So during the dedifferentiation, the cell will regain the capacity of division. So then comes dedifferentiation. Then comes the dedifferentiation, where the cell that has lost the capacity of division will regain the capacity of division. Where cells that have lost the capacity of cell division will again regain the capacity to divide. Remember, regain the capacity to divide. And this is under specialized condition, all right? So, like for example, interfascicular cambium, cord cambium, all these things. Example basically includes interfascicular and cord. All right. So that's why we can say the undividing parenchyma cell, they start to divide mitotically during the plant tissue culture. So option C is the correct answer. So differentiation cannot be the case because it will lose the ability of division. Redifferentiation, in case of redifferentiation, that comes just after the redifferentiation, cell will again lose the capacity of divide. So cells loses capacity of division. Capacity of division. So definitely this cannot be an option. And secondary growth is an overall process, all right, where the cell will be having the capacity of division, but only up to a certain time. So during the cell growth, during the secondary growth, all these processes are involved, not a single process. So definitely this cannot be the case. So that's why the correct answer is the D differentiation one. All right, now the next is question number 18. So question number 18 is a match the column kind of a question that has been given over here. So let us talk about it. So you have to choose the correct answer from the options given. So adenine. Adenine we all know is a nitrogenous base. All right. So this is a category of nitrogenous base belonging to purine category. So this is a purine. Adenine along with guanine is basically a purine. So it is purine which is found in the nucleic acid. Nucleic acid, that is DNA and RNA. All right, so this is going to match up with A. Then the second one is anthocyanin. Anthocyanin is basically considered to be as a pigment. And this is a category of secondary metabolite actually. So anthocyanin is a pigment or belonging to secondary metabolite category. Secondary metabolite. The C1 is chitin. Chitin is basically a complex polysaccharide which is made of NAM and NAG. Actually. So NAM actually, that means N-acetyl glucosamine. So chitin, it's a complex polysaccharide. This is a complex polysaccharide of N-acetyl glucosamine. Okay. Which is usually found in chitin cell, fungal cell wall. And the last is codeine, which is alkaloid. Codeine, and this is a category of alkaloid. This is also an example of secondary metabolite. All right. So let us take a look into the matched up options. So A is going to match up with fourth. Then B is going to match up with first. C is going to match up with second. And D is going to match up with third. So that means option B is the correct answer. Now question number 19. Question number 19 says that the residual persistent part which forms the perisperm in the seeds of beet is known as. All right. Now let me tell you that this question has been asked from the chapter of your sexual reproduction in flowering plants of standard 12. And the part, the remaining, residual part, which forms the perisperm in the seeds of the beet, that is known as nucellus. So nucellus is basically considered to be as persistent, or yeah, rather you can say perisperm is considered to be as persistent nucellus. That's why. So perisperm is known as persistent nucellus because in this case, then the some portions of the nucellus remains persistent, and that ultimately forms the seed. So persistent nucellus found in the seeds of wheat. All right, so that is the case, and that's what's called persistent nucellus. And the other example also includes, apart from wheat, the other example also includes black pepper. All right. And regarding integument, integument is basically the seed covering. These are seed coverings. So definitely this won't be an option. Calyx is basically, this basically represents sepal of flower. Flowers. Definitely this won't be an option. Endosperm is considered to be as that tissue of the seed, which is which is filled with reserve food material. So a tissue filled with reserve food material. Important. So definitely this won't be enough. Right. So nucellus is the correct answer. Option D is the correct answer. Now question number 20. Question number 20 says that the World Summit on Sustainable Development held in 2002 in Johannesburg, South Africa, pledged for. This question has been asked from the chapter of your basically biodiversity and its conservation of your standard 12 from your ecology section. So the World Summit that was held on sustainable development in 2002 in Johannesburg, so that pledged for collection and preservation of seeds of different genetic strains of commercially important plant. No, this cannot be the option, definitely. Then a significant reduction in the current rate of biodiversity losses. Yes, this is the correct answer, because the summit that was held in 2002, and particularly on the sustainable development, the main motive was to reduce the rates of the biodiversity losses that was occurring at a very high frequency. So option B is the correct answer. Declaration of more biodiversity hotspots, again, no. Increase in the agriculture production, again, no. All right. So we can say that the World Summit on Sustainable Development held in 2002 in Johannesburg, South Africa, with 190 countries, in which India was also a part of this summit, pledged their commitment to achieve by 2010 that there will be a significant reduction. There will be significant reduction in current rate of biodiversity loss at global, regional, and local levels. So at global, regional, and local levels. All right. So this was about the, you know, this was the main motive of the meeting that was, or the summit that was held in 2002 in Johannesburg, South Africa. All right. So that means option B is the correct answer. So let's talk about next question, which is question number 21. So this question says that the type of tissue which is commonly found in the fruit wall of nuts is sclerid, parenchyma, parenchyma, and sclerenchyma. Now this question has been asked from the chapter of your anatomy of flowering plants of standard 11. And let me tell you that this is a kind of a direct question that has been asked. Now, what is exactly these tissues? So, all these tissues that you can see, they belong to the complex permanent tissue, simple permanent tissue category. All right. So, like parenchyma, parenchyma, and sclerenchyma. These are all the simple permanent tissues. And out of these, the sclerenchyma is one such category of the tissue, simple permanent tissue, which is having two categorizations. So it is basically has two forms. One is called as sclerid and the other is called as fibers. So either they are available in the form of or present in the form of sclerids, or they are formed in the form of fibers. All right. Now, let me tell you that since this is asking, it is found in the fruit wall of nuts. So the nuts of fruit wall is basically made of dead tissue. All right. So since the, rather you can say, since the fruit wall of nuts is made of dead tissue, so in that respect or in that category, the parenchyma, parenchyma, and sclerenchyma that we talk about, the sclerenchyma is usually composed of dead tissues. Since this is made of dead tissues, therefore, the sclerid of sclerenchyma is found in fruit wall of nuts. And not only it is found in the fruit wall of nuts, but it is also found in case of pulp of fruits like guava, pear, and sapota. Then this is also found in case of seed coats of legumes like peas and beans, and also in the leaves of tea. So we can say that they are also found in, so they are also found in pulp of fruits like guava, pear, and sapota. Then seed coat of legumes, like peas and beans. So that's why leguminous plant species. Legumes and leaves. All right. So that's why the fruit wall of nuts is usually made of this claret part. All right. So that means option A is the correct answer. Now question number 22. Question number 22 says that the pioneer species in the hydrarch succession are, all right, filamentous algae, free floating angiosperms, submerged rooted plants, and then phytoplanktons. Now, this question has been asked from the chapter of your ecosystem of standard 12. And let me tell you that there are basically two types of succession. One is called hydrarch and the other is called xerarch succession. Now, what are pioneer species exactly? So pioneer species are considered to be as those species which will start the process of succession or which initiates the process of succession. So they are the first colonizing species, you can say. Colonizing species in an area, and that starts succession. This is actually going to start the process of succession. So that's why they are called pioneer species. All right. So in case of hydrarch succession, if you talk about, so the first category of plant species that will be considered to be the pioneer species are the phytoplanktons. All right. So we can say that in case, or rather in hydrarch succession, the pioneer species are phytoplankton. Because once they will be first colonizing the area, then they will be followed by, that means this hydrarch succession will be ultimately followed, this phytoplanktons will be ultimately followed by rooted submerged plants, or rather it will be followed by, then there will be the filamentous algae. Filamentous algae will be then carried out by the submerged rooted plants, and then free floating gymnosperms. So that is the case. So that's why the correct answer is option D. Now, question number 23. Question number 23 says that which of the following protects the nitrogenase inside the root nodule of a leguminous plant body? All right. Now, this question has been asked from the chapter of your mineral nutrition of your standard 12. And let me tell you that this is asking about the protection of the nitrogenase enzyme. All right. So nitrogenase enzyme is one such enzyme which is responsible or which is helpful in the nitrogen fixation. That means without which the entire nitrogen fixation is not possible. So let us first talk about nitrogenase. So nitrogenase is a nitrogen fixing enzyme. So this is basically a nitrogen fixing enzyme, and this nitrogen fixing enzyme is highly sensitive to molecular oxygen. All right. So which is sensitive to molecular oxygen. That means if there will be the presence of aerobic condition, then nitrogen fixation won't take place because the nitrogenase will get deactivated. All right. So that's what this nitrogen is a nitrogen fixing enzyme which is sensitive to molecular oxygen, and thus it requires complete anaerobic environment, or you can say anaerobic conditions to fix atmospheric nitrogen. All right. So how this anaerobic conditions are basically provided to the nitrogenase, or how this demand of anaerobic condition is fulfilled. So this demand of anaerobic condition is fulfilled in the nodules which are found in the roots of the leguminous plant. So you can say that in the root nodules of leguminous plants, there is a very peculiar structure which is being found, and that is called as leg hemoglobin. All right. So that leg hemoglobin, it contains naturally, that means these nodules, they contain an oxygen scavenger which is called as leg hemoglobin. So in the root nodules of leguminous plants, an oxygen scavenger called leg hemoglobin is found. So leg hemoglobin is found that traps the oxygen. So this is going to trap the oxygen and thereby ensuring that the enzyme is protected from the oxygen, and ensuring that enzyme is protected from oxygen. And when it will be protected from oxygen, so definitely the anaerobic condition is maintained, and so the nitrogenase is going to fix the atmospheric nitrogen. So this is how it is carried out. So let us take a look at the options that has been given. Glutamate dehydrogenase, no. Catalase, no. Leg hemoglobin, yes. Transaminase, no. All right. So these enzymes, that means glutamate dehydrogenase, catalase, or transaminase, they are other alternative enzymes for fixing other components of nitrogen, not the atmospheric nitrogen, but other components of nitrogen actually. So that's where the correct answer is option C over here. Now, question number 20. And question number 24 says that given below are the two statements, statement 1 and statement 2. And in the light of the above statements, you have to choose the correct answer from the options given below. So statement 1 says that DNA polymerases catalyzes polymerization only in one direction, that is 5' to 3'. All right. During DNA replication, or during, and the statement 2 says that during replication of DNA, on one strand, the replication is continuous, while on the other strand, it is discontinuous. Now, this question has been asked from the chapter of your molecular basis of inheritance of standard 12. All right. And looking into the statement, if you see, so both the statements are considered to be as the correct statement. All right. And the reason is that we can first write that both statements 1 and 2 are correct. 1 and 2. So they are considered to be correct. The reason being is this, that the first statement which says that DNA polymerases catalyze polymerization only in one direction, this is a true statement, because the enzyme which is responsible for catalyzing this process, it polymerizes or it catalyzes the polymerization of DNA only in one direction. So statement 1 all right, which says that. So we can say that DNA polymerases, these are basically the enzyme that catalyzes the polymerization or synthesis of DNA. Catalyzes synthesis or polymerization of DNA in only one direction. And that is 5 prime to 3 prime. So this is a correct statement. All right. Now the statement 2, which says that during replication of DNA, on one strand the replication is continuous, while on the other strand it is considered to be as discontinuous. This is again also true. The statement is also true because the direction is same. All right. So the replication on one strand is considered to be continuous, while the on the other strand it is considered to be as discontinuous, because there is, in continuous strand of the DNA, there is a continuous synthesis. Only one, one RNA primer is required for carrying out the process of DNA replication. While in case of the strand which is discontinuous, multiple RNA primers are required, and Okazaki fragments are formed, and that's why it is discontinuous. So, statement second is also correct. So during replication of DNA, one strand, or rather you can say on one strand, the replication is continuous. The replication is continuous as it requires only one prime. As it requires only one RNA primer to, along with other nucleotides, to complete the entire synthesis. And nucleotides to complete synthesis. While the replication on the other strand is discontinuous due to formation of several Okazaki fragments. So Okazaki fragments are nothing but they are the DNA strands only, or DNA fragments only. Okazaki fragments in the direction of 5 prime to 3 prime, requiring multiple RNA primers. All right. So this is the reason. So that's why both the statement are considered to be as the correct statement. That means option B is the correct answer. All right. Now, the next is question number 25. Question number 25 says that the species that comes to appear in bare area are called species of cereal community, pioneer species, invasive species, and competitive species. Now, this is a question that has been asked from the chapter of your ecosystem, and that is again a very direct question that has been put forward in front of you. So, the species, the very first important species that colonizes a bare area, that is known as the pioneer species. All right. So, the first species that colonizes a bare area all right, is termed as pioneer species. That is considered to be a pioneer species. Like, for example, if there is a Zerach succession. So, in case of Zerach succession, the pioneer species are lichens, right? So, you can say that in Zerach succession, that means the succession which is going to start in the dry areas. That is called Zerach succession. The pioneer species are lichens on rocks. All right. While in case of hydrart succession, if you see, the pioneer species are phytoplanktons. All right. So this is the case. So that's why the correct answer is option B over. All right. Now, the next is question number 26. So question number 26 says that initiation of lateral roots and vascular cambium during the secondary growth, it takes place in the cell of. All right. So pericycle, epiblema, cortex, and endodermis. So this question has been asked from the chapter of your anatomy of flowering plants of standard 11. And let me tell you that if there is initiation of lateral roots and vascular cambium during the process of secondary growth, it is asking about. All right. So if you just look into the sequence, so it basically starts with anatomy, basically starts with epiblema, which is also called as epidermis. So the sequence of tissue basically lies in the fact, sequence of plant tissues, if you just write it. So it starts with epiblema, which is also known as epidermis. Then it will be followed by cortex. All right. So cortex can either be parenchymatous or hypodermis or anything, whatever that can be the case. Then the innermost cortex is called as endodermis. And then it will be followed by pericycle, single layered, which is usually single layered. Pericycle is followed by vascular bundles. All right. So this pericycle will be ultimately followed by vascular bundle consisting of xylem and phloem. Vascular bundle with xylem and phloem and cambium in between. All right. So the last tissue, if you just see, the roots are being initiated and the vascular cambium has to be formed. All right. So vascular bundle with xylem and phloem and cambium in between, you can say. Xylem. All right. So the last tissue to be present over here is only the pericycle that you can see. Right. So you can see the pericycle over here. So it is the primary tissue of the plant body that is going to give rise to lateral roots and the secondary meristems. That means they will be giving rise to the vascular cambium and the cord cambium, which is basically the phyllogy. All right. So we can write this part that pericycle is the site of initiation of lateral roots and vascular cambium and cord cambium. All right. So that's why the correct answer is going to be the pericycle. Option A. Now, the next question is question number 27. So question number 27 is a match the column kind of a question that has been given. All right. So basically, in this question, match the column kind of question has been asked. So basically, if you just look into the options that has been given, so this is a question that has been asked from the Lac-Operon section. That means this question has been asked from the chapter of your molecular basis of inheritance from the Lac-Operon part. All right. So let us take a look into the different genes that has been given. Talk about the different options. So the first option, which says that in Lac-Operon, the I gene is basically responsible for. So I gene is one such gene. Basically, there are one repressor gene, you can say, and there are three structural genes. All right. So this I gene is basically a repressor gene. All right. And inducible gene that you can say. And this I gene is responsible for encoding for a repressor protein. All right. So that is the case. So this is basically a regulator gene. Regulator gene. That means I gene. All right. So this codes for repressor protein. All right. So that's why the correct answer here is option. Now, the second is Z gene. Now, as I told you that there are three structural genes. So Z, Y, and A, these are all three are the structural genes. So the Z gene basically encodes for beta galactosidase. So codes for beta galactosidase. So this beta galactosidase is basically responsible for cleaving the lactose into glucose and monoamines. All right. So that is the case. Now the third one is, this will go with B, is Y gene. Y gene is going to encode for permease. All right. So Y gene encodes for permease. That means it will increase the permeability of the cell for the uptake of lactose. So that's why it encodes for permease enzyme. And the last one is the A gene. So A gene is going to encode for transacetylase. All right. So A gene encodes for transacetylase. All right. And apart from that, there are an operator gene and a promoter gene as well. So it hasn't been asked over here, so we will only look into that part. So let us take a look at the options that has been given. So A is going to match up with fourth. This one. B is going to match up with third. Then C is going to match up with second, and D is going to match up with one. That means option C is the correct answer. All right. Now, question number 20. Question number 28 says that to ensure that only the desired pollens fall on the stigma in artificial hybridization process, uh, the female flower buds of plant producing unisexual flowers needs to be bagged. Then there is no need to emasculate unisexual flowers of selected female parent. Emasculated flowers are to be bagged immediately after cross-pollination, and emasculated flowers are to be bagged after removal of anthers. Bisexual flowers or bisexual flowers showing protogyny are never selected for cross. All right. So out of all the options that that has been given below, you have to choose the correct answer. All right. So in this, well, now let me tell you that this question has been asked from the chapter of your sexual reproduction in flowering plants of standard 12. All right. So while in case of artificial hybridization process, so if there is a need or a desire to focus on this part only, that only the desired pollen grains are going to fall on the stigma during the artificial hybridization, there are two things which are required. One is called as the emasculate, and that too in a bisexual flower I'm talking about. There are two processes of artificial hybridization. Let me tell you first. So in artificial hybridization, there are two processes in a bisexual. That means if you want to dust your desired pollen grains, your type of pollen grains onto the flower. That's why. So there are two processes in a bisexual flower. So the first one is called as emasculation. All right. So emasculation is basically the removal of anthers from the floral tip before they dehisce to release the pollen grain. So this is basically the removal of anthers from flower tips using a pair of forceps before they dehisce to release anther. All right. So this is about the emasculation. So in case of emasculation, both the things will be, in case of bisexual flower, both the methods are carried out. That means emasculation, along with that, bagging will also be done. So bagging is usually done in those flowers which are emasculated. All right. So emasculated flowers flowers are bagged with a suitable cover. So they are being covered, they are being bagged with a suitable cover, and it is only done to prevent the undesirable cross pollination. So it is done to prevent landing of undesirable pollen from cross pollination. All right. So once the bagging is carried out, so these two methods are done in case of uh, bisexual flower. But if there is unisexual flower, so in case of unisexual flower, there is only bagging which is required. So in unisexual flowers, only bagging is required, no emasculation. All right. So if you just look into the options that has been given, so the first option says that the female flower buds of plant producing unisexual flowers needs not to be bagged. Again, this is a wrong statement because we need to bag the, as I told you, that in case of unisexual flower, the bagging is required because until and unless to prevent the landing of undesired pollen, prevent landing of undesired. So if you do not want the undesired pollen grains to land onto the stigma of the unisexual flower, so the bagging is must. Bagging is must. So definitely this is the wrong statement. Now the next statement says that there is no need to emasculate the unisexual flowers of the selected female parent. Yes, true. This is a true statement because here only I have written that in unisexual flowers only bagging is required to prevent the landing of the egg. No emasculation is done. So there is no emasculation. All right. So no emasculation needs to be done. This is true. Then this third statement says that emasculated flowers are to be bagged immediately after cross pollination. Yes, this is again very, very important that once the bagging is done, the next step that you need to do is that you will be dusting the once the stigma gets matured. So once stigma is matured, stigma is matured, the desired pollen grains are dusted onto stigma. Pollens are dusted onto stigma. And once the dusting is done, once the dusting has been done, then the rebagging needs to be there until and unless the fertilization has occurred successfully. Now, after rebagging, the tagging is done. All right. So definitely, this emasculated flowers needs to be bagged. So this is also the correct statement. Then emasculated flowers are to be bagged after removal of anthers of the bisexual flowers showing protogyny are never selected. Bisexual flowers showing protogyny are never selected for cross-pollination. So this is also considered to be as correct. Protogyny is basically a condition where the gynoecium will be maturing. That means the female reproductive organ will be maturing earlier than the male reproductive organ. So definitely in case of bisexual flowers, we need to select those flowers that is not showing protogyny. They are never selected for the cross pollination. All right, because if the gynoecium will be maturing earlier, then it will be very difficult to prevent the cross colon landing. All right. So that's why this statement is also correct. So if you just look into the options that has been given, so it's B, C, and D, which is correct. So A, D, and E, A, B, and C, B, C, and D. That means option C is the correct answer. All right. Now, the next is question number 29. So question number 29 says that the ascent of xylem sap in plants is mainly accomplished by the root pressure, size of the stomatal apparatus, distribution of stomata on the upper and lower epidermis, cohesion and adhesion between the water molecules. So this question has been asked from the chapter of your transportation in plants of standard 11. All right. Now let me tell you that the ascent of xylem sap is basically the movement of the water along with the mineral nutrients it is asking about. Ascent of xylem sap is basically this word or this term basically means the movement of the water along with the mineral nutrients. So that forms the ascent of xylem sap. So ascent of xylem sap is, and this ascent of xylem sap is primarily related with two important phenomena that is worked out in the water molecule, or that is found in the water molecule, and that is called as cohesion and adhesion actually. All right, so we will be discussing this as well. The water and mineral nutrients movement. Nutrients movement upward the plant. So upward the plant body. So this process, that means the ascent of xylem sap is mainly triggered by transpiration. So it is triggered by transpiration process only. This is triggered by transpiration, but it is actually dependent on two important factors. See, ascent of xylem sap has nothing to do with the root pressure, because root pressure, which is developed because of the root pressure, the water won't be taken up towards the upward plant of the upward parts of the plant body, but will only be restricted in the root region, because the root pressure is very very less. So it is not responsible for ascent of xylem sap. So ascent of xylem sap is mainly triggered by this phenomenon, of your you can say transpiration, which is called as transpirational pull theory. All right. So size of the stomatal apparature, it has nothing to do with size of the stomatal apparature, distribution of stomata on the upper and lower epidermis. So it actually depends upon two physical properties of water. So it depends on physical properties. So the first property is known as cohesion. Cohesion is basically a kind of a mutual attraction between the water molecules. That is called as cohesion. So, mutual attraction between water molecules. All right. So, this is the case. And the other one is the adhesion. Adhesion is considered to be as a mutual interaction between water molecules and the surface which is carrying that particular water molecule. That means the polar surfaces. So adhesion, the attraction of water molecules to polar surfaces. Because of these two factors only, the water is able to develop an upward driving force along with the mineral nutrient, and so it goes to it, and so it is carried to the uh, from roots to the upper parts of the plant. All right. So that's why the correct answer is cohesion and adhesion between the water. All right. Now, the next is question number 30. So question number 30 says that this is basically a match the column kind of a question that has been asked. So you have to find it out. You have to match the column with regard to the different kind of estivation. So basically, if you just look into the list that has been given, so it is basically the estivation that has been mentioned. That means imbricate, volvate, maxillary and twisted, and calotropis, cassia, cotton, and wheat. So this question has been asked from the chapter of your morphology of flowering plants of standard 11. All right. So estivation is something where how the leaves, how the petals or sepals are arranged in the floral bud. So the pattern of arrangement of sepals or petals in a floral bud with respect to one another, that is called estivation. So we can just define estivation. So it is the pattern of arrangement So it is the pattern of arrangement of sepals or petals in floral bud with respect to one another. All right. So that is the case. Now there are various kinds. They can be volvate, they can be imbricate, they can be vexillary, or they can be either twisted. All right. So let us take a look into the different categories. So the very first important category is considered to be as the volvate category. So volvate estivation is one such estivation where all the sepal or petal that is found in a floral bud, they are aligned in an equal manner without touching it. All right, so that is the case. So it basically looks like this when you just take a look. So this is going to give an appearance like. So it's going to go like. This is like volvate, like they are just touching their edges. All right, so this kind of arrangement is basically found in, you can see that this kind of arrangement is found in calotropis. All right, now the next one is considered to be as the imbricate estivation. So imbricate estivation All right. So imbricate estimation again is one such estimation which basically follows a pattern of 1 plus 2 plus 2, you can say, or rather there is a pattern which is being followed, and that is known as 1 plus 1 plus 3 pattern which is being followed. That means one will be completely inside. That means one wall will be completely inside, one will be completely outside. So this will be inside, this will be completely outside, and three whorls will be like this, then they will be inside and outside. So one edge will be inside and one edge will be outside. So one edge inside and one edge outside, like this. All right. So if we just draw it, so we can just like, so this is going to go like this. Like this. So you can see this is completely inside. This is the one which is completely inside. Then this wall is completely outside. Both its edges are, outside. And then this one is inside. This one, this is belonging to, these are the edges which you can see over here. Let me show you. Yeah, which you can see. These three are the one. These three are the one whose wall, which is having one edge outside and one edge inside. So these three are the walls which you can see over here. Inside, this is inside, outside, inside, outside, inside, outside. So this is how it looks like. All right. So that is the case in case of imbricate estivation. Then the third category, and yes, the imbricate estivation is particularly found in Cassia and Gulmohar. So this is found in Cassia and Gulmohar. All right. While the next is basically the twisted estivation. Now, this twisted estivation basically means that all its floral whorls will be arranged, in a twisted manner. So, twisted manner basically means that the arrangement will be something like this. All right, so this is something which is twisted. So you can see that every edge is like one edge is an inside and the other edge is outside. So that's how the thing will go like. So this kind of twisted estivation is usually gonna go for rose, then cotton, and lady's finger. All right, so that is the case. And the last is considered to be as vexillary estivation. So vexillary again is a very unique estivation. Vexillary estivation, if we talk about. So vexillary is also known as papilionaceous estivation, where you will see a bigger, you know, bigger wall like this. Then inside it, you will find inner two walls like this. And inside it, you will again find two inner walls. And this is joined together. All right. So this is called as auxiliary activation actually, and this is called as keel. All right. So the largest petal is basically considered to be a standard. So this is the standard petal which you can see over here. All right. Covering the two anterior petals, two smaller lateral petals which are called wing. So this is called wing, and this is called keel. Correct. And this is found in case of peas and beans. That means all the leguminous plant species. All right. Now, let us take a look into the options now. So, imbricate has been found, as we have seen over here, that imbricate has been found in cassia. This one goes with A. Volvate, as usual, it has been found with calotropis, which is with B over here. Auxiliary is found with bean, and twisted is found with cotton. All right. That means B option. So let us take a look into the matching options that has been given. So A is going to match up with second. Okay. Then B is going to match up with first. Then C is going to match up with fourth. And D is going to match up with third. All right. So that's why the correct answer is option C over here. All right. So the next question is question number 31, which says that the number of times the decarboxylation of isocitrate occurs during the single TCA cycle is 4, 1, 2, and 3. All right. So this question has been asked from the chapter of your respiration in plants of your standard 11. And let me tell you that during the entire TCA cycle, which is the tricarboxylic acid cycle, there are basically uh, two steps where the decarboxylation takes place. All right. So there are two steps of decarboxylation. There are two steps of decarboxylation. All right. So the very first important step is lies in the fact that it lies in basically in the fourth step of the entire TCA cycle. So during the fourth step or at fourth step, you can say when there is conversion of isocitrate into or decarboxylation of the isocitrate into alpha ketoglutarate. So during fourth step, there is decarboxylation of isocitrate to, and this isocitrate is a 6 carbon molecule. So it will be decarboxylated to form a 5 carbon product, 5 carbon product, which is alpha ketoglutarate. Alpha ketogluteric acid, you can say. All right, so this is the first step of decarboxylation where the CO2 is released. And the second step, the decarboxylation occurs at the fifth step. All right, so just succeedingly to this step, at the fifth step itself, this alpha ketoglutarate, which is formed over here, which is a 5 carbon molecule, that will now be undergoing the decarboxylation to form the succinyl COA, you can say. So alpha ketoglutarate which is a 5 carbon molecule undergoes decarboxylation to form a 4-carbon product. So this will be forming 4-carbon product, and that is succinyl CoA. All right. So at these two steps, the decarboxylation is taking place over here. But here, since in the question it is asking about the number of times the decarboxylation of isocitrate is occurring during single TCA cycle. So it will be only one. So at only one step, the isocitrate is getting decarboxylated to form a 5-carbon product, which is alpha ketoglutric acid. So the answer will be option B over here. All right. Now, the next is question number 32. So question number 32 is the match the column kind of a question. So list 1 and list 2 has been given. So let us take a look at the different options. So the first is basically the porins. Now porins are basically a category of a very specialized proteins. It's a type of a specialized proteins which is responsible for forming pores in the outer membrane of mitochondria, so that the movement of molecules will be easy. All right. So we can say the first option is that porins. So porins are basically, it's a type of protein that forms huge pores in outer membrane of mitochondria. So with the help of porin proteins, ultimately the molecules, since it will create pores, so ultimately the molecules will be having very ease to move from the extracellular environment to the intracellular environment. All right, so that is the case. Now option B is leg hemoglobin. Now leg hemoglobin is basically a hemoglobin which is found in the root nodules of the leguminous plants, and because of the color of basically the leg hemoglobin is usually pinkish in color. All right, so this leg hemoglobin acts as an oxygen scavenger and thereby fixing nitrogen. So leg hemoglobin, so it is found in root nodules of leguminous plants. So it is specifically found in the root nodules of leguminous plants and is pink in color. And acting as oxygen scavenger. As O2 scavenger. Thereby fixing nitrogen. All right. So this is the case. So option, so this is going to match up with B option. All right. Now the next is proton accumulation. Now proton accumulation occurs in the lumen part of the thylakoid of chloroplast. All right. So the proton pumping occurs from the, basically from the outside section of the chloroplast inside the lumen. So that means the stroma, the pumping usually takes place from the stroma towards the lumen of the thylakoid. That means inside space of the thylakoid. So we can say that proton accumulation occurs in lumen of thylakoid of chloroplast. All right. Now, the last is D1 and respiration. So respiration is considered to be as an amphibolic pathway because it involves both anabolic and catabolic processes. So that's why it's considered to be as an amphibolic pathway. So respiration, this is an amphibolic pathway. As it involves both anabolic and catabolic processes. All right, so that is the case. So let us take a look at what are the matching options. So A is going to match up with fourth, B is going to match up with first, then C is going to match up with second, D is going to match up with three. So option C is the correct answer. Now, question number 33. Question number 33 says that separation of DNA fragments is done by a technique known as gel electrophoresis, polymerase chain reaction, recombinant technology, and southern blotting. So let me tell you that this question has been asked from the chapter of your biotechnology principles and processes of standard 12. And again, this is a very direct question, definitive question that has been asked. So the process by which the separation of the DNA fragments, that means the cutter DNA fragments, will take place. So that method is called as gel electrophoresis. All right. So you can say that gel electrophoresis is a method of separating DNA fragments Now, this separation of DNA fragment basically occurs on the basis of their molecular size. So, on basis of molecular size of DNA, size of DNA fragments under the effect of electric field. Under under the effect of electric field. So this is the correct answer. That means gel electrophoresis is the correct answer. All right. While the other, options that has been given, so polymerase chain reaction, if you talk about it, so this is basically a process of amplifying DNA fragments. So process of amplification of DNA. That means if you need to make a multiple copies of a certain DNA segment, then that process will be used. So PCR is called as amplification. It's a process of amplification of DNA segment or a fragment, you can say. All right. Then recombinant technology. Recombinant technology is basically the production of recombinant DNA. It's a technology which is based on producing recombinant DNA. So technology based on production of recombinant DNA, which is in shortly termed as rDNA. While on the other hand, southern blotting technique is a technique of transferring the RNA molecules. Not the RNA, but the DNA molecules onto a blot paper. So So basically there are three techniques: Western blotting, Southern blotting, and Northern blot. Western, Southern, and Northern blotting. So that's why. So transfer, so this is a technique of transfer of DNA, or rather you can say separated DNA, separated DNA fragments onto a blotting paper. That can either be a cellophane paper or a nitrocellulose membrane, basically. So blotting paper. All right. So that's why the correct answer is gel electrophoresis, option A over here. All right. Now question number 34. Question number 34 says that in general, the egg apparatus of embryo sac in the angiosperm consists of. So this question has been asked from the chapter of your sexual reproduction in flowering plants of standard 12. And for knowing this answer, let us first draw the structure of embryo sac. So a typical angiospermic embryo sac is usually oval in shape, if you just see to it. And it is basically having two ends. It's having a closure like this. Like this. Then there is now. So as I told you, that a typical angiospermic embryo sac is having two ends. So one is called as the challegal end. So this is basically the challegal end of the embryo sac, and this is known as the micropylar end. Challegal end and micropylar end. And a typical angiospermic embryo sac is usually made of, it is usually made of 7-celled and 8 nucleate. Embryo sac is 7-celled and 8 nucleate. Now, when I say 7-celled and 8 nucleate, so this primarily means that so this is the embryo sac that we have drawn. All right. So these are what? These are considered to be as antipodal cells. These are antipodal cells which are 3 in number. This is basically the central cell. So central cell is basically having 2 polar nuclei. So this inside which you can see is the polar nuclei. So this, if you consider cell, it is 1, but if you consider nuclei, then they are 2 in number. All right. Then this becomes the egg cell, which is again single in number. And these are basically the synergids. All right, which is 2 in number. So if you just calculate, so it is 7-celled and 8 nucleate. So cells are 7, but if you just consider the nucleus, so 3 antipodal cells, 1 central cell, then 1 egg cell and 2 synergids. So 2, 3, then 4, 5, 6, 7. But if you consider the nucleus state, then 3, 4, 5, 6, 7, 8. All right. So this is basically the nucleate condition that you have to consider. You don't have to consider in this part. All right. Central cell. So 7 cells, and 8 nucleate. So let us take a look. So it is 1XL, 2 synergids, 2 antipodal cells, 2 polar nuclei. Again, wrong. 1XL, here there will be 3 antipodal cells, not 2 antipodal cells. 1XL, 2 synergids, 3 antipodal cells, and 2 polar nuclei. Okay, this has been true. This is considered to be true. But let's take a look in the other options as well. 1XL, 2 synergids, 2 antipodal cells, and 3 polar nuclei. Again, no. 1XL, 3 synergids, 2 antipodal cells, and 2 polar nuclei. Again, this is wrong. And the last is not applicable one. So again, this is also wrong. So the correct answer is basically the option B over here, which is 1XL, 2 synergids. You can see 1XL, there are 2 synergids, then 3 antipodal cells, and 2 polar nuclei that you can see over here. All right. So that's why option B is the correct answer. Now, question number 35. So the floral diagram represents which one of the following families? It is asking about it. So basically the floral diagram is missing over here. So we cannot deal with the, question for this part. So let us just skip this question. Then question number 36. Question number 36 says that the primary proteins, they are also called polypeptide because they can assume many conformation. They are linear chains. They are polymers of peptide monomers. Successive amino acids are joined by peptide bonds. Now, this question has been asked from the chapter of your biomolecules of standard 11th. Now, let me tell you that primary proteins, they are called as polypeptide because they are the, linear proteins which are joined together, or linear amino acids which are joined together by peptide bond. So, primary proteins are called polypeptides. So they are called polypeptides because they have amino acids which are joined together or joined by peptide bond. All right, so let us draw this part as well. So if you just draw it, normal, structure. So this basically goes like this. Like this. So we will be first drawing, I'm first drawing up the peptide bond so that you people can have an idea. Again, this is going to be like this. So this is the first amino acid, which you can see. This is the first amino acid. All right. Now, so amino acid is basically represented by this. Now, we can draw again the another amino acid. Right. Correct. So this is the second amino acid. All right. So if we just, so basically peptide bond is formed as a result of dehydration process between 2 amino acids. So here the elimination of water occurs. That means the process of dehydration takes place between 2 amino acids, and then only the peptide bond will be formed. So if we are just going to eliminate this water part from here, as you can see, so what will be the bond that will be formed, and this will connect the 2 amino acids. All right. So how the structure goes, so it will be like this. All right. So this, which you can see, this bond is considered to be as peptide bond. Right. So we can see from this figure basically that the dehydration process has taken place and the two amino acids, they are joined together by this peptide bond, which is also known as amide bond. This is basically an amide bond. All right. So definitely the options that has been given. So it is, they are called as polypeptide. Reason being is this, primary proteins, because the successive amino acids, they are joined together by the peptide bonds. So that means option D is the correct answer. All right. Now, question number 37. So question number 37 says, this is a match the column question that has been given. So regarding the different number of base pairs and nucleotides are found in different categories of organisms. So let us take a look. So the very first important is category is the bacteriophage, which is 5x174. So bacteriophage 5x174 virus. This is basically a virus. So this virus has the presence of, this is a very direct question that has been asked, because all the figures are already given in your book. All right, and this is a question that has been asked from the chapter of your molecular basis of inheritance of standard 12. So in case of the bacteriophage 5x174, the single stranded DNA has the presence of 5,386 nucleotides. That means this one. So the single stranded DNA has 5,386 nucleotides. All right. Now, option B is bacteriophage lambda. Bacteriophage lambda is basically, they have the presence of 48,502 base pairs. All right. So there they have linear DNA. So their linear DNA basically has 4,502 base pairs. So bacteriophage lambda. So they have 48,502 base pairs. All right. Now, the third one is Escherichia coli. Escherichia coli have the presence of Escherichia coli, they have circular DNA actually. All right. So since they have circular DNA, so that's why they have the presence of 4.6 into 10 to the power 6 base pairs. So E. coli have circular DNA, and their circular DNA has the presence of with 4.6 into 10 to the power 6 base pairs. All right. And the last one is haploid content of human DNA. So the haploid content of human DNA basically means one complete set of chromosomes. So this is basically 3.3 into 10 to the power 9 base pairs. So haploid content of human DNA. So this is 3.3 into 10 to the power 9 base pair. All right. So that means C is going to match up with 4 and D is going to match up with 4. So let us take a look into the options that has been given. A is going to match up with second. A is going to match up with second. B is going to match up with first. This one. C is going to match up with fourth and D is going to match up with third. So that's why option D is the correct answer. All right. Now, the next is question number 38. So question number 38 says that which type of substance would face difficulty to pass through the cell membrane? Substance soluble in lipids, substance with hydrophobic moiety, substance with hydrophilic moiety, all substance irrespective of hydrophobic and hydrophilic moiety. Now, this question has been asked from the chapter of your transportation in plans of standard 11. Now, let me tell you that plasma membrane, which is also known as cell membrane. So, plasma or cell membrane is basically made of phospholipid bilayer. Is composed of phospholipid bilayer. All right. So plasma membrane is typically made of phospholipid bilayer. And since this is made of lipid layer, so this phospholipid bilayer is usually, this is usually of hydrophobic nature. Hydrophobic nature. That means the inner side is hydrophobic, but the outer side is considered to be as hydrophilic. But since the molecule has to pass through, the molecule has to diffuse through the cell membrane. So for diffusion, it is very, very necessary that the molecule should pass the lipid bilayer. So if the molecule should be soluble in lipid, so the diffusion will be very easy. So substances soluble in lipids, their diffusion will be easy. So here diffusion will be easy. So diffusion is easy. Why? Because the membrane is itself made of the substance is itself made of lipid, and the membrane is also made of lipid. So it is very easy. But substances with hydrophobic moiety, again, this is here also the diffusion is considered to be easy because, as I told you, that it is usually of hydrophobic nature over here. All right. Now, substances with hydrophilic moiety, this here the diffusion is going to be difficult. All right. So that means here there is difficulty in diffusion. There is difficulty in diffusion. And the reason is that since lipid bilayer is made of, it is a hydrophobic layer or a hydrophobic or of hydrophobic nature. So the molecules which are of hydrophilic nature, like water and all, they are passed across the cell membrane through specialized channels and specialized carrier proteins. All right. So they cannot easily pass through. So you can say that substances which are hydrophilic can't be easily passed through membranes. Membrane. So they actually require specialized carrier and channel. So here the difficulty of diffusion is very difficult. Diffusion is very difficult. So that's why the correct answer is going to be C only. But let us take a look at the option. So basically, the D statement, which says that all substance, irrespective of hydrophobic and hydrophilic moiety, again, this is also a contradictory statement, so this cannot be correct. So that's why the correct answer is option C over here. All right. Now, the next is question number 39. So question number 39 says that what is the expected percentage of the F2 progeny with yellow and inflated pod in a dihybrid cross experiment involving pea plants with green colored inflated pod and yellow colored constricted pod? All right. So this is a question that has been asked from the chapter of your principles of inheritance and variation of standard 12. And let me tell you, so we'll need to draw a cross. So if we are going to go with the cross, so the cross is green color pods with inflated pods. So green and inflated pods. This we are going to cross with yellow and constricted pod. So yellow and constricted pods. All right. So this is basically the dominant character. That means we know that green color pod and inflated pods, they are the dominant character. So definitely in case of the F1 generation, we will be getting all the dominant character. So in F1 generation, these are basically the parents, you can say. So in F1 generation, you will be getting all green and inflated pods. All green inflated pods. All right. While in case of the F2 generation, technically, if you just have done the dihybrid cross also, so we get a ratio of 9 is to 3 is to 3 is to 1. This is the phenotypic ratio that we get it. Now we have to find out what? We have to find out the genotypic ratio. This is the phenotypic ratio, but we need to find out the genotypic ratio. So this 9 is to 3 is to 3 is to 1, the ratio that we get it basically belongs to 9 basically belongs to green and inflated. That means the very dominant character. All right. Then 3 basically belongs to green and constricted. Green and constricted. All right. Then again, 3 will be belonging to yellow and inflated. And the last one will be completely recessive. So it will be yellow and constricted. Yellow and constricted. All right. So we know that basically here it is asking about that we need to find out the expected percentage of F2 progeny with yellow and inflated pods. All right. With yellow and inflated pods, we need to find it out in a dihybrid cross experiment involving pea plants with green colored inflated pod and yellow colored constricted pod. So we need to find out this one. All right. The genotypic or the expected progeny of this. So the total, how you're going to find it? So this is basically the recombinant. All right. So the F2 progeny for yellow inflated pods. So that is 3 divided by total number, and the total number is 16. 9 plus 3 plus 3 plus 1 will be 16. So this will give out, an aggregation percentage of, if you just find it out, then it will give out a percentage of 18.75%. All right. So that's why if you just look at the options, option D is the correct answer. All right. Now, the next is question number 40. The question number 40 says that, again, this is a match the column kind of a question that has been given, list 1 and list 2. So you need to match it up. So option A state carbon dissolved, that is related to 55 billion tons, 71%, 4 into 10 to the power 3 kgs. Then there is 2 into 10 to the power 10%. Now let me tell you that total number of carbon which is found dissolved in the entire ocean is 71%. So carbon dissolved in oceans, you can say. And this question is basically related with, this entire question is basically related with the ecosystem chapter of your, standard 12. So carbon dissolved in oceans, the percentage of carbon dissolved in oceans is 71%. So this is A. Now, annual fixation of carbon through photosynthesis is basically the entire fixation, annual fixation, if you talk about it, according to an estimate that has been given. So that is totally 4 into 10 to the power 3 kgs of, you know, uh, carbon is fixed annually. So annually, if you say, then 4 into 10 to the power 3 kgs of carbon is fixed by plants through photosynthesis. All right. So that is the case. This is going to match up with B. Then power captured by plants. So if you see the power, photosynthetically active radiation, which is captured by plant, it is only 2 to 10%. So out of the total 50% of the incident solar radiation, plants can utilize only 50%. But 50% of the incident solar radiation that reaches the earth, out of those 50% solar incident solar radiation, the plants, they capture only 2 to 10% of the light, so which is called as photosynthetically active radiation. So photosynthetically active radiation, or rather in short you can say PAR, is 2 to 10% of 50% incident solar radiation. Solar radiation. So this is 2 to 10% of the 50% incident solar radiation. That means C option. The last is productivity of oceans, which is approximately around 55 billion tons. And this productivity is low because of the availability of low nitrogen and organic nutrients. So productivity, though the oceans, they cover a major area of the entire globe, but the productivity contribution by oceans is less. So productivity of oceans is 55 billion tons. All right, so that means this is the D option. Now let us match it. A is going to match up with second. A is going to match up with second. Then B is going to match up with third. B is going to match up with third. That means this one. C is going to match up with fourth. And D is going to match up with first. Right. So this is, that means option D is the correct answer. All right. The next is question number 41. So it states that if a female individual is with small round head, furrowed tongue, partially open mouth, and broad palm with characteristic palm trees, also the physical, psychomotor, and mental development is retarded, the karyotype analysis of such an individual will show. So let me tell you that this question has been asked from the chapter of your principles of inheritance and variation of standard 12. And the options that is being followed is trisomy of 21, so 47 chromosomes with XXY sex chromosomes, 45 chromosomes with XO sex chromosomes, and 47 chromosomes with XYY sex chromosomes. All right. Now, the symptoms that has been shown in the above question, like small round head, furrowed tongue, partially open mouth, and broad palm. So this is all the characteristic features of Down syndrome. All right. So basically, the Down syndrome is one such syndrome in which there is a trisomy condition. So this Down syndrome is caused due to trisomy of 21st chromosome. So this is usually caused because of the trisomy of 21st chromosome, you can say. All right. And this trisomic condition, that means an additional set of chromosome, if you will talk about it, trisomy. So this will result into 2n plus 1 chromosome. So this is a classic case of aneuploidy. This is a category or a type of aneuploidy. So 2n plus 1, this represents aneuploidy condition. Aneuploidy. And aneuploidy is basically, it is caused either because of there is an additional, there is an extra chromosome or an additional chromosome that is added to the former set of the chromosome, or it is either the deletion of the chromosome. So here the trisomic condition is there. So definitely there is an additional. So this indicates that there is an additional one chromosome in the 21st chromosome. So this indicates the presence of an additional chromosome at 21st chromosome. There is an additional chromosome at the 21st chromosome. All right. And all these symptoms is caused. That means the symptoms with small round head, furrowed tongue, partial open mouth, these are all seen in case of Down syndrome. So the symptoms include, so Down syndrome symptoms are, so the symptoms are small round head, then there is furrowed tongue, then there is palm crease, in broad palm, then physical and mental retardation, etc. So these are the symptoms of Down syndrome that we have seen. Broad palm with palm crease, physical and mental retardation, all these things. So that's why the correct answer here is option 1. All right. And these options, which is 47 chromosomes with XXY sex chromosomes, so this is a case of Klinefelter syndrome. So this is Klinefelter's syndrome. Then 45 chromosomes with XO condition. So this is a case of classic case of Down syndrome. Then 47 chromosomes with XYY sex chromosome. Again, this is also a case of Klinefelter. All right. So this is the case. That's why the correct answer is option A over here. So the next question is question number 42. So this question says that you have to read the following statements and identify the characters related to the elega shown in the diagram. So the diagram has been represented below and certain characteristic features have been given and you have to identify the correct statement. All right. Now, from the above diagram, which you can see over here, this diagram is basically a representation of chapter 3rd, that means plant kingdom of standard 11th. All right. So this is a figure of a green elega, which is Cara. So this is the figure of a green elega, which is Cara, which is a green elega. All right. So it's a member of Chlorophyceae basically. All right. So definitely it's a member of chlorophyce. All right. So since this is a member of green alga, so definitely this is a member of chlorophyce. So that means option A is the correct statement. Then the second statement says that food is stored in the form of starch. So this is also true because A is actually correct. Then the option B is also correct. Reason being is this, that in case of green algae or chlorophyce, the food is usually stored in the form of starch. So food is stored in the form of starch in green algae. So in all the members of the green algae, the food is actually stored in the form of starch. All right. Now, the option C says that it is a monoecious plant showing oogonium and enthelidium. Again, this statement is also considered to be correct. This is also correct. And the reason is that monoecious plant means a plant which will be bearing both the male and the female reproductive structure, which is enthelidium and oogonium. All right. So it's a monoecious plant. So this is a monoecious plant bearing the male, which is male reproductive structure. So bearing the male reproductive structure, which is entheridium, and female reproductive structure, and female reproductive structure, which is oogonium in the same plant. So this is also correct. All right. Now the third statement says that food is stored in the form of mannitol and laminarin. Now this is incorrect because the food is stored in the form of laminarin in brown algae. So this is actually wrong. All right. This is not the correct answer. Then it shows dominance of pigments, which is chlorophyll A, C, and fucoxanthin. Again, this is also being shown in case of brown algae. So this is also a wrong statement because the pigments which are found in case of this chlorophyce, that means Cara, is chlorophyll A and B, just like to that of the green plants. All right. So from the above options, we can say that A, B, and C is the correct answer, correct statements. So if you just look into the option C is the correct answer. All right. Question number 43. Question number 43 says that the list 1 and list 2 has been given and you have to choose the correct answer from the options given below. All right. So let us take a look that the different options that has been given. Now the first is sacred groves. Basically, sacred groves are a category of in situ method of conservation. All right. So this is one such category of in situ method of cultivation, or sorry, conservation, where it includes the tract of the forest which is conserved by the people because of some religious and cultural beliefs. All right, thereby emphasizing the in situ conservation. So one of the areas where sacred groves are found is the Khasi Hills in the Meghalaya. So this is going to be the correct answer. That means option. So sacred groves are one of the methods of in situ conservation. This is actually one of the methods of in situ conservation where large tracts of forest they are conserved by people due to religious and cultural beliefs. All right. So one of the areas, the example where the sacred grove is found is the Khasi Hills of Meghalaya. All right. So that's what the correct answer is option A over here. Now, the second is the zoological park. Zoological park is a method of ex situ conservation where the animals are protected. All right. That means the animals which are taken in or taken out from their natural habitat and are in turn protected in their own by creating a you know, by creating a specific habitual conditions. So that's why it is a method of ex situ conservation. So the B option is zoological park. So it is a method of it is basically a method of ex situ conservation where threatened animals are taken out of their natural habitat and given care and protection. All right. So that's why this one. The Nile perch. Nile perch is basically one of the examples that represents alien species. That means an exotic species, all right, that was introduced in the Lake Victoria. And it ultimately led, this is Nile perch is actually a category of fish. So this was introduced in the Lake Victoria that ultimately led to the extinction of more than 200 species of cichlid fish, which was the native fish of that lake. All right. So we can say that option C is Nile perch. So an alien species. Introduced in Lake Victoria that led to extinction of more than 200 species of native cichlid fish. All right. So this is the case. And the last is Amazon forest. So Amazon forest is basically related with release of large quantity of oxygen because it covers approximately more than 14% of Earth's land surface. So that's why it covers, or it is responsible for releasing the largest amount of oxygen. So the option D is Amazon forest. It covers more than 14% of Earth's land area and releases large amount of oxygen. All right. So this is going to match up with D. So let us take a look into the concerned options that has been given. So A is going to match up with 4th. Okay. Then B is going to match up with 3rd one. Then C is going to match up with 1st. And D is going to match up with 2nd. So that's why the correct answer is option B over. All right. The next is question number 44. So question number 44 says that the enzyme A is needed for isolating the genetic material from the plant cells, and enzyme B is used for isolating the genetic material from the fungus. Now choose the correct pair of options from the following. All right. So this question has been asked from the chapter of biotechnology principles and processes, where you might have studied it also, that for isolating the genetic material, certain specific enzymes are used for treating specific cells. Like for example, if you have to isolate the genetic material from the plant cell, then you have to treat it with a specific different enzyme. So we can say that for isolating the genetic material, so for isolating the genetic material from plant cells, the enzyme needed is cellulase. Enzyme needed is cellulase, because we know that the plant cell is composed of cellulose. Cell wall is composed of cellulose, so that's why we need to treat it with cellulase enzyme. All right. As plant cell wall is made of cellulose. All right. Now, for isolating the genetic material from fungal cell, and for fungal cell, the enzyme needed is chitinase. The enzyme needed is particularly the chitinase, because chitin is the composition of the cell wall of fungus. As fungal cell wall is made of chitin. That's all. All right. So that means cellulase and chitinase will be the option. So let us take a look. Cellulase, lipase, cellulase protease, cellulase chitinase. That means option C is the correct answer. All right. So lipase is basically used for removing the fats. All right. While the protease is used for treating or for isolating the proteins from the cell. All right. So that's why option C is the correct answer. Now, question number 45. Question number 45 says that Identify the correct sequence of events during prophase 1 of meiosis 1. All right. So synapses of homologous chromosomes. Chromosomes becomes gradually visible under microscope, crossing over between the non-sister chromatids of homologous chromosome, terminalization of chiasma, and dissolution of synaptonymal complex. All right. So during the process of out of all the options actually that has been given over here, so the very first thing that we need to know that we have to focus on the prophase 1 of meiosis 1. So prophase 1 of meiosis 1. All right, so it's prophase 1 of meiosis 1 actually. So in this process of prophase 1 of meiosis 1, if you just look into it, then the very first process that will be there is the visibility of the chromosomes during the process when they will start condensing themselves. All right. So that's why option B will be the very first one. That means when the chromosomes becomes gradually visible under the microscope, and this occurs during the process of leptoty. All right. So the very first stage is B1. That means chromosomes becomes gradually visible under microscope. So this is an event which occurs during the leptoteen phase. So this occurs during leptoteen phase. That's why this is the first one. Now, so B will be the first option. Then there will be the second option that will be there is the once these chromosomes will be visible, then the process of bivalent formation will take place, and the bivalent will only form when the process of synapses will occur. All right. So that's why when you look into the options, this one, that means synapses of homologous chromosome, this is the second one. So that means the second one sequence is synapses of homologous chromosomes. So synapses of homologous chromosomes will result into bivalent formation. Results into bivalent formation, and this occurs during the zygotine phase or zygotine process. All right. Now, the next is basically the once the bivalent has been formed, the next step is the process of crossing over to occur between the non-sister chromatids of homologous chromosome, and this occurs during the Pecitine stage. So the next is C1. That means crossing over between non-sister chromatids of two homologous chromosomes. All right. So this is the case. And the crossing over between the non-sister chromatids of two homologous chromosomes, this will occur during Pecitine stage. All right. Then the fourth process to take place is primarily the once the synapse, the process of this, that means your this one has taken place. So this crossing, once the crossing over has taken place, so this will result into exchange of the genetic material. That means the recombinants will be formed, and once the recombinants will be formed, then there has to be the dissolution of the synaptonymal complex. That means the point has to be dissolved at the point of crossing over. All right, so the next is Dissolution of synaptolimal complex after the process of crossing over. Synaptolimal complex. So this occurs during the process of diploating, because the paired homologous chromosomes will now start moving apart. All right. And the last is left with the D1, which is terminalization of chiasma. So the D1 is basically the terminalization of chiasma. That means once the dissolution of the synapses or synaptolimal complex has taken place, the X-shaped structure is formed at the point of the crossing over, which is called chiasma. So that has to be terminalized. The terminalization of chiasmata that results into X-shaped structure. Of chromosome. And this occurs in the last process, which is dikinesis. All right, this is the case. So if you just look into the options, so B, A, C, E, D is the sequence. So B, A, C, E, D. That means option D is the correct answer that you can see. All right. Now, the next is question number 46. The question number 46 says that which of the following pair represents the free-living nitrogen-fixing aerobic bacteria? Now, this question has been asked from the chapter of your mineral nutrition and from the section of the nitrogen fixation of your standard 11. So let us categorize the different categories that has been given. So in case of free-living nitrogen-fixing bacteria, so free-living nitrogen-fixing bacteria, they have been divided into two categories. Either they can be aerobic or either they can be anaerobic. So free-living nitrogen-fixing bacteria. So they are of two types. They can either be aerobic, anaerobic, all right. So let us first talk about the aerobic. Aerobic categories of free-living nitrogen-fixing bacteria includes Azotobacter, then Azospirillum, then there is Biserinckia, and then there is Dirksia. These are the four most important category of aerobic nitrogen free-living organism. While in case of anaerobic, if you just like consider it, then in case of anaerobic, there are, Rhodopseudomonas and Bacillus categories are also there. This includes Rhodopseudomonas. All right. So if you just look into the different categories that has been given, so Pseudomonas and Theobacillus. Pseudomonas and Theobacillus, these are actually the denitrifying bacteria. All right. So these are not the nitrogen fixing bacteria, but these are denitrifying bacteria. Both are denitrifying bacteria. All right. Then there is Rhizobium and Biserinckia. Rhizobium is a nitrogen fixing, symbiotic nitrogen fixing bacteria. Again, this cannot be the option. So Rhizobium, it's a symbiotic nitrogen fixing bacteria. All right, this is the case. And Biserinckia, we all know, is an aerobic category. We have seen. So again, this won't be the option. Then Azotobacter and Biserinckia, yes, this has been mentioned. So this is the correct answer. And the last is Anabina and Rhodospirillum. Again, Anabina is a nitrogen. This is a free living also, but it is a nitrogen category also that comes over here. So again, and Rhodospirillum is a category of facultative bacteria. So again, this won't be an option. So that's why the correct answer is option C over here. All right. Now, the next is question number 47. Question number 47 says that the frugivorous birds are found in large numbers in the tropical forest, mainly because of, all right, so it is temperature conducive for their breeding, lack of niche specialization, higher rainfall, availability of fruits throughout the year. All right. So let me tell you that the frugivorous birds are those birds that eats fruits. All right, and they are largely found in tropical forest because we all have studied that in the chapter of that biodiversity and its conservation of standard 12, that tropical areas, they are the less disturbed areas, less seasonal areas, and more constant and predictable, and that is ultimately going to promote niche specialization and greater species diversity. All right, as compared to the temperate one. So if you just look into all the options that has been given, so it is the availability of fruits throughout the year, because of which they are found largely in the tropical forest. All right. So we can say that frugivorous birds are fruit eating birds. That are largely found in tropical forest, as tropical areas are less seasonal, more constant, and predictable, unlike temperate ones. So they actually are going to promote greater species diversification. So they promote niche specialization and greater species diversity. All right, that's why. So that's why the correct answer, if you just look into the options that has been given, is the availability of fruits throughout the year. Since the season is very constant, it is not very fluctuating. So that's why there is availability of fruits all throughout the year. Option D is the correct answer. Question number 48. Question number 48 says that identify the correct statement regarding the chemoosmotic hypothesis. All right. So you have to find out the correct statement regarding the chemoosmotic hypothesis. And this question has been asked from the chapter of your photosynthesis in higher plant of standard 11. All right. Now, chemoosmotic hypothesis is basically related with the production of ATP in the cell organelles. All right. So if you just look into the options that has been given, that is splitting of water molecule takes place on the inner side of the membrane. Exactly, this is a true statement, because until and unless the water molecule will be splitting, the protons won't be released and the proton gradient won't be created. So definitely this is a correct statement that takes place on the inner side of the membrane, which is the stroma, which is the lumen part of the this one, of the lumen part of the thylakoids. So option A is the correct answer, you can say. Splitting of water molecule releases protons, thereby creating proton gradient or thereby forming proton gradient on inner side of membrane. All right, so this is a correct statement. Again, the protons accumulate within the lumen of the thylakoid. This is also true. The accumulation of protons will occur on the lumen of the thylakoid. Then only the proton gradient will be developed, and these protons will be pumped from lumen to the stroma of the thylakoids. So yes, since the proton gradient is developed, so proton gradient, or rather you can say protons are accumulated inside lumen of thylakoids. All right. And this creates proton gradient. Gradient from lumen to stroma. All right. So this is also correct. Now, primary acceptor of electron transfers the electrons to an electron carrier. There is nothing like the process of transferring of electron that happens in case of this proton, in this chemiosmotic hypothesis. So definitely C is not involved, because this is a process which is involved in ATP. This is a process which is involved in triggering the ATP synthesis, but not exactly in the ATP synthesis. So this we cannot consider it. Then NADP reductase enzyme is localized on the stroma side of the membrane. Again, this is true. Yes, the NADP reductase enzyme is present on the stromal side of the membrane. And the last is protons increases in number in the stroma. Since the protons, they are released and accumulated in the lumen, so the number is high in the lumen and lesser in the stroma. So the pumping takes place from the lumen to stroma. So again, this option also cannot be considered to be as a correct one. All right, because the gradient, if it would have been asked about the gradient, then yes, the gradient is developed towards stroma. But since it is asking about the proton number, increment in the proton number, so definitely it is in the lumen, not in the stroma. All right, so the D option is also correct. Is also correct, right? Which states that the NADP reductase enzyme is localized on the stromal side of the membrane. So if you just see, so A, B, and D is the correct one. So option C is the correct answer. Now, question number 49. Question number 49 says that the list 1 and list 2 has been given, and you have to identify the and match it up with the correct answer from the options given below. All right, so this question has been asked from the chapter of your biotechnology principles and processes of standard 12. Now, gene gun. Gene gun is basically a method which is also known as biolistics. All right, so gene gun is a biolistics method in which there is a process of gene transfer. All right, so gene gun or biolistics, which is also known as biolistics, is a method of DNA transfer in a plant cell. All right, so definitely this is going to go with A. Then there is gene therapy. Gene therapy is a method by which any faulty gene is replaced by a normal healthy gene. And one of the classic examples that we have seen is the ADA deaminase deficiency enzyme, or ADA deaminase deficiency that we have seen in a 4-year-old girl. All right. So gene therapy. So it is the replacement of a faulty gene by a normal healthy gene. And one of the best example is ADA deficiency in 4-year-old girl. All right. So the next is gene cloning. Again, gene cloning, as the word is suggesting, cloning is there. So definitely, it's the method by which identical copies of the particular DNA molecule is obtained. All right. So C is gene cloning. The process to obtain identical multiple copies. So identical multiple copies of DNA. All right. Then the last is genome. So genome is basically the total amount of DNA that is found in the cell of an organism. All right. So genome is the total amount of DNA present in cells. All right, so this is the case. So if you just match up with the options that has been given, so A is going to match up with second, then B is going to match up with first, C is going to match up with fourth, and D is going to match up with third. All right, so that means option D is the correct answer. Now, question number 50. Question number 50 says that which of the following can be expected if scientists succeed in introducing apomictic gene varieties of crops? All right. So this question has been asked from the chapter of your sexual reproduction in flowering plants of standard 12. All right. And here it is asking about the apomixis process. So the first option is that there will be segregation of the desired characters only in the progeny. Polyembryony will be seen and each seed will produce many plantlets. Seeds of hybrid plants will show longer dormancy and farmers can keep on using the seeds produced by the hybrid to raise new crop year after year. All right. Now, out of all the options that has been given, the correct answer is option D over here, because apomixis is one such process where the seeds are produced without fertilization. All right. So apomixis is a process of producing seeds without fertilization. All right. And this has also resulted into hybrid seed formation. All right. So this apomixis, the plants which are produced as a result of apomixis, it is actually identical to the parent plant. All right, because there is no kind of fertilization is involved. So the plant produced are hybrid plants. Produced are hybrid plants. All right, that means they will be identical only to their parent. So it is one such advantage that the farmers need not have to buy the seeds year after year. Rather, they can keep on using the seeds produced by the hybrids to raise new crop every year. So we can say that farmers can keep on using apomictic seeds year after year. All right. So that is the uniqueness. That's why option D is the correct answer.